Welcome, everybody, and welcome back to the Biodiversity Conference 2021, particularly in the vertebrate section on new discoveries coming out of the Vertebrate Genomes projects and in collaborative efforts. And I think we have a nice, good lineup of speakers. We have Yafe Mao from Washington University, Ines Miranda from University of Porto, Eliza Ramos from the University of Campinas in Brazil, and Simona Secondi from University of Milan. And finally, we'll end off with Marcelo Uliana de Silva and Vladimir, I don't want to butcher his name, Ochinskas from Sanger. So you'll hear him pronounce it correctly. And I think, there's wonderful research that's coming out of all these vertebrate genomes that the group is producing. And really, this is the very first time we have a session really talking about only the biology as opposed to the assemblies. So what we're going to do is hear all the talks one after another. And there's a chat box you can enter your questions in. Please go ahead and enter your questions as they come to your mind, as opposed to waiting to the end, or you can wait till the end. And I have my co-chair here, Rong Rei, who many of you know, Rong Rei. And do you want to say something to the group?
Yeah, welcome everyone. And I'm very happy to see that we have finally more to talk about the genomes, including the one that we presented last year. And yeah, today we have very diverse set of speakers, which are presenting very diverse set of vertebrate genomes as well. Yeah, and once again, as a reminder, please go ahead and don't be shy to put in any questions that you want to ask the speakers. I'm sure that if you have a question in mind, someone else will have the same question in mind too. So it's not going to be only you. So yeah, please go ahead and type in any questions that you have.
Good, all right, great, thank you. So we'll see you on the other side.
Okay. Thanks to organizers for giving me this opportunity to share my work with you. Today, I'm going to talk about the structural variation and in-community sorting in greater apes. The greater apes include orangutan, gorilla, chimpanzee, bonobo, and human. Chimpanzee and bonobo are sister species belonging to the pangenus. They are split from 1 million years ago. This unique system provides a great opportunity for us to study the hominid speciation. Understanding the evolution of the greater apes could help us to address a very fundamental and important question: what makes us uniquely human, and what makes a champ as a champ? In the evolution of the greater apes, mutations are the original source to the genetic variation. Genetic variation include in SMV, indel, and structural variation have been diversely accumulated in each lineage. And also, the different evolutionary forces have been shaping in the genome of the greater apes, in particular in community sorting and the natural selection. Therefore, I'm interested in two questions here. First, what is the genome landscape of structural variants and in-community sorting among greater apes? Second, how do structural variants and in-community sorting driven by the natural selection lead to adaptation in greater apes? However, as a result of lower accuracy of next generation sequencing, large proportion of the greater ape genome cannot be compared. Luckily, in past few years, the long-read technology have enhanced our ability to generate more contiguous genomes. The ECLO lab have been working on the greater ape genome project for decades. In 2016, our lab generated the first de novo long-read genome assembling of gorilla and continue working on the other greater ape genomes. After I joined the lab, I used a different long-read technology to assemble the bonobo genome and to study the structural variation and in-community sorting in greater apes. I use long-read mapping and the genome alignment to discover structural variation in bonobo. And you see here, the blue represents the insertion, the red represents the deletion. And then I use the population data to genotype this structural variation. And as you see, most of the structural variants are polymorphic here. Then I apply this approach to other great apps and build the phylogeny of the fixed structural variation in great apps. In total, we discovered more than 60,000 novel fixed structural variants in great apps. And the 17 protein coding genes have been disrupted by the fixed structural variants. And more than 1,000 structural variants intersect with the encoded regulatory elements. Here is an example about the EIF483. I used the Hi-Fi rates to resolve the genome structure of this gene and found 6 copies of the EIF483 in bonobo and 5 copies in CHAMP. And this is a pangenous specific duplication. And then I used the 4-length isoform RNA-seq to show 4 copies are expressed in bonobo lineage. Then I reconstructed the evolutionary history of EIF483. As you see here, this duplication occurred at a common ancestor of the bonobo and the CHAMP, and then independently duplicated in the bonobo and CHAMP lineage. We also observed the several amino acid changes in these duplicated genes. We also use a different fish experiments to validate these duplication events. The EIF483 is a major component of the axon junction complex. It is associated with neuron developmental disease. So it would be very interesting to look at the gene function in bonobo and the CHAMP in future. The other example is about the human-specific gene RGPD6. By comparing the human genome and the non-human primate genome, I found a human-specific region which contains RGPD6, the Q duplicon. And this human-specific region provides a substrate for the inversion and the deletion of the NPHP1 gene. The deletion of the NPHP1 gene is associated with the human disease, the Gerbert syndrome. Beyond the unique variation in each lineage, I'm also interested in the shared variation among great apps. The previous short-read genome assembling, NinCan analyzed around 800 megabase ortholog region among great apps. And they found around 1.6% of the human genome is more close to bonobo compared to chimpanzee. And proposed this IRS region is associated with the long-term balance selection, like MHC region. However, with the advance of the long-read technology, we now can analyze around 2.4 gigabase of the human genome. And we found around 2.5% of the human genome is more close to bonobo compared to chimpanzee. Then we project this IRS on the human genome. We observe that some of the IRS seem like to be clustered together. Then we did a simulation and we found around 21% of IRS is more likely to be clustered together than non-distribution. More interestingly, the axons near the IRS sites have a higher DNDS value, implying the relaxed or the positive selection on the IRS sites. Further, we examined the population diversity in chimpanzee and the bonobo, and we found the clustered IRS sites have a lower pi diversity compared to the non-clustered IRS site in chimp population. Also, we found the same pattern in bonobo population. In summary, the long-read sequencing have discovered more than 60,000 previously unknown fixed structure variants, which could distinguish the human and the pine lineage. The long-read sequencing increased by the twofold the proportion of the IRS, and the subset of the IRS is not randomly distributed in the greater epigenes. Third, the different selective forces may drive subset of the IRS in the evolution of the greater apps. Thanks to my advisor, Dr. Evan Eckler, and our collaborators. Thanks for your listening.
Hello, everyone. My name is Ines, and today I will talk to you about our work on the evolution of seasonal coat color variation in leaf tweezers. Starting with some background context, seasonal color change is a key adaptive trait that consists of the alternation between summer brown coats and winter white coats. This trait is shared by several mammal and bird species, and it's vital to the maintenance of their camouflage in environments with seasonal snow cover. If animals are mismatched, for example, having turned white when there is no snow on the ground, as you can see in these two photographs, they become highly visible, increasing their predation rates and decreasing their fitness. These mismatches are associated with snow cover decreases that are driven by climate change. And given the predictions of further snow cover reductions, the survival of these populations is expected to be increasingly endangered. Importantly, many of these species have polymorphism of the winter coat color, with some populations that remain brown throughout winter. And the distribution of both winter morphs has been shown to correlate with the extent of snow cover across the species distribution range. And as such, when considering the fact that winter white phenotypes are expected to undergo strong selective pressures in a context of climate change, the fact that we have this standing genetic variation associated with this polymorphism may allow rapid adaptation to the changing climates. In this work, we studied the evolution of this polymorphism in the leaf tweezel, which is one of the species with two alternative winter phenotypes. This species occurs throughout the Holarctic, and in Europe, both phenotypes exist. And in this map, you can see the distribution of both winter morphs, with the white morph occurring in the north and the brown morph occurring in the south. The intermediate gray areas that you can see are what we call transition zones between color morphs and are areas where both phenotypes coexist. For this work, our sampling was based exclusively on specimens from natural history collections, and we targeted two transition zones between winter colors, one in Sweden and one in Poland. And we sequenced the genome of several specimens at low coverage, resulting in a total of 79 whole genomes. And in addition, we also produced a new reference genome for this species using an opportunistically sampled road-killed weasel. With this data, we tried to answer three main questions. The first being, what mechanisms explain the maintenance of the two winter morphs? The second, which genes are responsible for the winter polymorphism? And the third, what is the adaptive value of this color variation? And so focusing on our first question, we looked into the genetic structure of the populations and assessed its correlation with color morph distribution. And in these two graphics, you can see on the left our admixture plot, where the phenotype of each specimen is shown by the colored circles. And in the right, you can see a population tree inferred based on allele frequencies. And what the results show in both analyses is that the primary axis explaining the genetic differentiation between the animals is the geographic origin. And so this suggests that the winter color morph distribution is not associated with the underlying population structure, but instead it's likely driven by local selective pressures. Next, to investigate the genetic basis of this polymorphism, we conducted whole genome scans of association where we compared allele frequencies of alternative winter phenotypes. And here you can see our case control association test, where only one significant peak was identified, and this is shown by the yellow rectangle. And when we look further into this genomic region, we see that it includes 7 protein coding genes. So to further explore this region and to validate this association, we took advantage of a previous crossing experiment that had shown that the winter brown morph was the dominant phenotype. So we used the genotyping assay with SNPs covering the entirety of the association region, and we genotyped animals that were produced in the crossing experiments and that we also sampled in a museum collection. And the results of our genotyping that you can see here show that the winter white morph is homozygous for one of the alleles, while the winter brown morph is either heterozygous or homozygous for the alternative allele. Then we extended the genotyping to both natural populations of Sweden and Poland. And in Sweden, we see that both phenotypes again show the expected genotyping pattern following the dominance of the brown. And in Poland, we found that the same significant association between genotype and phenotype exists, but we see that half of the specimens that are highlighted here by this red rectangle have a haplotypic block with highly reduced genotype association. And this allowed us to identify two SNPs that are perfectly associated with the color morph across specimens and that fall in the MC1R gene. And this is quite striking because MC1R is a very well-known pigmentation gene that is involved in the melanin production pathway. Its interaction with agouti, that is another gene from the pathway, it's fundamental to determine the color of the pigment that is deposited in the skin, which can be light or dark pigment. And notably, the AZIP gene has already been associated with winter color polymorphism in two hare species, the snowshoe hare and the mountain hares. And as such, together with our results, this suggests that we have the same pathway being repeatedly recruited to generate winter color polymorphism across species, but through different genes. Finally, we also ran some selection scans to look for signatures of positive selection that could hint at the adaptive value of the trait. And our results that you can see here in these plots identified a selective sweep overlapping the MC1R region that is shown by the gray shading in the brown morph, but not in the white morph. And these results suggest that the brown alleles were favored at some point in the past, possibly in response to local environmental selective pressures. And this can hint at their potential to help adaptation to climate change and also to help rescue endangered winter white populations in the near future. So to sum up, our results show that the distribution of the polymorphism is driven by local selective pressures. We have identified MC1R as a strong candidate gene associated with the polymorphism, supporting the repeated recruitment of the same pathway across species. And we have evidence of positive selection that hints at the adaptive value of the winter brown alleles. To end, I would just like to thank all the people and institutions that made this work possible. Thank you all for your attention, and I'm happy to take questions.
Hi, everyone. My name is Elisa, and I'm excited to be here today to talk about how we use high-quality genomes and comparative genomic approaches to explore the unique and shared genomic footprints of two species of sea turtles. Currently, there are only 7 species of sea turtles in our oceans, and we are particularly interested in the leatherback turtle, which is Dermochelis coriacea, and the green sea turtles, which is the Cheloniamides. These two turtles belong to different families inside the sea turtle lineage, which diverged from each other around 60 million years ago. Separated by many years, those two species accumulate several physiological and morphological traits that differentiate them, and also present different distributions around the globe, with the leatherback turtles being able to navigate to more colder waters in high latitudes. Nowadays, we have access to powerful genomics tools that could help us to understand the evolution and help in the conservation of these threatened animals. However, till now, only a draft genome for the green turtle is available on public databases. And although the draft genome offered a lot of information to explore questions about turtle evolution, that genome is still highly fragmented. And unfortunately, till now, there was no highly quality resource available for any other species of sea turtles. The good news is that our team is trying to fill this gap and led by Lisa Komorowski and Camila Mazon, we generated and explored high-quality genomes for the sea turtles. And with the help of an amazing group of collaborators, we are working to use this genomic data to describe the genomic uniqueness of sea turtles, their evolutionary story, and the molecular basis of their most interesting adaptations. We started assembling reference genomes for the leatherback and the green turtles, based on the vertebrate genome project pipelines. The two genomes assemblies produced show high levels of contiguity and completeness, and a complete annotation based on species-specific transcriptome data. Then, using this data, we explore structural and functional variation, comparing the two genomes from each other. Our first findings with the comparisons was quite surprising. We identify an extremely high overall synchrony between them, and we found a pattern of high identity throughout the entire genomes, represented here for the almost perfect straight line showing the matches between each one of the 28 chromosomes in a comparison to from leatherback to Chelonia. This high synchrony pattern was also observed for the functional part of the genome on both assemblies, as shown here, with the lines representing the connection between the orthogonal genes on the different assemblies. Also, both sea turtles present similar levels of repetitive elements on their genomes, around 42% for the leatherback and 41% of the entire genome for the green turtle. Therefore, we know that these species are very different in their ecology, morphology, and physiology. So where are the differences that make them unique? To find those differences, we first take a deep look on 14 chromosomes that exhibit a lower synchrony pattern in specific subregions, marked here in red on the dot plots. Those regions were compared with the surrounding regions for us to have a clue of what could be the reasons for the low identity between them. So what we found, here we are seeing the scaffold number 1, showing the two regions with low synchrony marked in red. We identify the function of the genes present in the entire chromosome, as well as the functions of the genes present on the low synchrony regions. In those low synchrony regions, we found multiple copies of genes related to the immune system and olfactory receptors for the green sea turtle when compared with the leatherback turtle. Also, we explored the presence of transposable elements in those regions, comparing with the surroundings, represented here in red and blue, while the low synchrony region is represented in yellow. The region present in low synchrony did not show a higher amount of transposable elements. However, it still presents a higher proportion of masked base pairs in comparison with the surroundings. That could mean that the transposable elements present in the region could be in fact longer than the transposable elements present in the surroundings. And that's exactly what we found, as we found average of length is slightly longer on the low synchrony region for these transposable elements. Those same patterns are found for almost all chromosomes that present regions of low synchrony, presenting more copies of immune system genes or olfactory receptors on the green turtle, combined with the presence of longer transposable elements associated to that regions. One exception to the pattern could be found in chromosome 20. Despite having a huge rearrangement region, there is no presence of immune or olfactory genes duplicated here. However, both sea turtles present the double amount of transposable elements in that region when compared with the surrounding regions. The chromosome 12 is also an exception. Although presenting a large inversion on the beginning of the chromosome, this region does not have any duplicated genes for both the species, and no presence of longer or higher number of transposable elements were found here. So with this first in-depth comparison between the two genomes, we are able to describe some patterns that could be associated with the region and diversification of the species. As for example, the bigger number of gene copies related to the king systems on the green turtle, that could be explained by an expansion of these gene families on the Cheloniid family or only for the green turtle, or even a loss of these gene copies for the Demochelid family. We also identify association of the presence of longer transposable elements with regions with high levels of gene duplication for the green turtle. And that's it by now. I would like to thank you all for watching, and thanks to all people and agents involved in this work. Those are my contacts, and I'll be glad to answer any questions if you have.
Hello, everyone. We are Simona Secomandi and Guido Gallo from the University of Milan. And our work is focused on the banzuolo, a small pestering bird that has been the focus of a huge amount of behavioral, ecological, and evolutionary studies. However, the lack of a chromosome-level assembly hindered the investigation of its genomics. We therefore generated a new reference genome for the species using the VGP standard pipeline 1.6 with subsequent manual curation. Some of the data were used from a previous banzuolo assembly generated by our group, which was more fragmented and scaffold level. And we also added newly generated data to reach the chromosome level. We were able to identify 39 chromosomes plus the sex chromosomes, and we also generated the first NCBI annotation for the species. We reconstructed the karyotype for the species, and we confirmed the chromosome number and also their size in our assembly. Those are the main characteristics of the reference genome. In particular, we confirmed the microchromosomes are GC-rich, CPG island rich, and gene-rich. The last 4 tracks refer to a reference-free multiple genome alignment performed with cactus using our assembly, 6 other passeriformed genomes, and also the latest chicken genome. And we performed with this alignment selection analysis with both PhilOP and FastCount. Those are the results from the PhilOP analysis, the accelerated and conserved size after FDR correction and also Bonferroni correction in the first Manhattan plot. And we are currently looking inside those peaks and inside those sites to find the list of candidate genes which are under selection. Just for completeness, we also performed the conservation analysis with FastCount. And in general, all the selected bases fell mostly into intronic and intergenic regions, while looking at the features themselves, the coding sequences were the most conserved. We also generated the first pangenome for the European banzuolo using the cactus pangenome pipeline. It involved both haplotypes of our VGP assembly and also primary and alternate Hi5-based assemblies of 5 banzuolo individuals. This is a portion of the variation graph in chromosome 1 of our assembly, which is the black stripe. And as you can see, it is possible to look at conserved regions, NIPs, but also more complex structural variants. The core genome between the sequences shared between the primaries were 90.5%. And also looking at the genes of our annotation, the vast majority is shared between all the individuals, while only 160 genes were not found either in the Hi5 assemblies nor in the Hi5 reads, meaning that they can be real gene losses in those individuals, but also being assembly artifacts or maybe absent for the fact that Hi5 reads have some difficulties in covering GR-rich regions. To explore even more the potentialities of Hi5 reads, we also performed a phasing experiment by aligning our Hi reads to our assembly, call the variants, and generate those very big blocks of phasing. And these are also the percentage of the phased chromosomes. Now, Guido Gallo will continue the presentation.
So the next step was to use our new reference genome to align our Hi5 data as well as all the genomic data that are publicly available for the species to generate a comprehensive catalog of SNP markers for the banzuolo. Here you can see a map of the sampling locations of different individuals used in the catalog, the work they refer to, as well as the type of genomic data. We evaluated SNP density across all banzuolo chromosomes. Here you can see as example chromosome 2 and chromosome 3, and we correlated this information with different genomic features such as repeat content and GC content. Since there are no studies in literature about linkage equilibrium in the banzuolo, we also used the SNP data from our catalog to estimate the genome-wide linkage equilibrium decay trend in these species using all data from different populations of the available data set. Here you can see the names of the Barzola subspecies of different populations. We saw that in general, LD decreases very rapidly, already within the first 10 kb. And we also saw that the highest values of LD were computed from populations with the smallest sample sizes, and this is in line with what observed in literature previously. Despite the very low values of LD at the genome-wide level, we were still able to isolate some regions exhibiting high values of pairwise LD between markers. In particular, one region on chromosome 6 harboring 4 genes, among which we identified BDNF as the most interesting candidate. In particular, the highest values of LD within this gene were computed from the Egyptian Barzola population, as you can see here from the heat map of LD values between genetic markers within the gene for this particular population. So BDNF was our top candidate, and we isolated it because of its involvement in neuronal plasticity and differentiation. Moreover, for the fact that in zebrafinch it is involved in strong learning, and it was also found that its expression is regulated through a change in the methylation status of specific CpG sites that are present in the regulatory region of the gene. Here in this picture, you can see that particular region in the Carpus multi-alignment that Simona explained you before, among the different bird species, where we also highlighted the CpG site as well as the SNPs in our catalog. As you can see, there is a high degree of conservation of these sites among birds, and in particular in the Barzuolo, 4 of the 6 sites that you see here are conserved as CpG sites. And one SNP in our Barzuolo catalog directly affects a zebrafinch methylation site. So to conclude, we generated a chromosome level assembly and a preliminary pangenome for the European Barzuolo. We also scanned the reference genome for signature of selection using a cactus alignment. We used our new chromosome level assembly and all publicly available data to generate a comprehensive catalog for the species. We also investigated genome-wide linkage equilibrium trend as reference information for future studies. And we also performed a genome scan to identify candidate loci in high linkage equilibrium. We identified BDNF as the most interesting candidate. In fact, it is involved in different aspects that are all related to tameness also. And this paves the way for future studies regarding this gene aimed at elucidating the evolution of the synanthropic habits of the Barzuolo. We are about to submit this work, so it will soon be available on bioRxiv. And we would like to thank also everyone who contributed in this project. And thank you for your attention.
Hi, everyone. Thanks for having us here. I am Marcela. Vi and I are going to be presenting you 3 Caecilia genomes that we have sequenced to a high quality and the common route to limblessness in vertebrates. So Caecilia are amphibians. We have around 214 extant species, and most importantly, they are limbless. They are tropical species. Here we have 3 Caecilia from 3 different families. They are about 100 million years diverged from each other. We have sequenced these 3 species genomes to a high quality with the Vertebrate Genomes Project and the Sanger part of the VGP. As you can see from the heat maps here, the Hi-C heat maps, they are very well assembled, especially after manual curation. Caecilia genomes are large, as amphibian genomes usually are, so they range from 3.7 gigabases to 5.3 gigabases in size, and we have put most of the base pairs, so more than 97% of all of the base pairs to chromosomes, which stands for a high quality genomes. These 3 Caecilia have very repetitive genomes, this large genome, so at least 67% of the genomes are composed of repeats. We have class 1 TAs, heterotransposons being the most prevalent, and they occur more than 20 times, are more abundant than class 2 TAs, the DNA retrotransposons. So there are many theories on how large genomes come to be and how they are maintained. And one of the theories would be that maybe the accumulation of all of these repeats is due to the lack of molecular control of a specific TA element. Nevertheless, among amphibians, if we consider salamander genomes and also frog and toads, they do have different types of TAs that accumulate in their genomes. So we would say for at least amphibia in general, it doesn't seem to be that it's only a failure of keeping one kind of TA in check. Other mechanisms must be involved. We have done first synteny analysis of predicted Busco genes with the Caecilia and with the common frog and toad, just to have a look at chromosome synteny. So here you can see that at least based on the Busco genes for the three Caecilia, we have synteny for the mostly for the three larger chromosomes, and then they are actually very not conserved in terms of the Busco genes. I mean, they are 100 million years apart, so we can see there is not a lot of conservation in terms of Busco gene syntenies with the Sicilian genomes. So these genomes were then sequenced with the VGP, curated, sent to the NCBI, so annotation was done there. And then all of the analysis of genes that we have done were done by Vlad V, with this these predictions of genes.
To study changes in protein coding part of Sicilian genomes, we collected data from RevSec and Ensemble for 21 species. Altogether, we got 400,000 genes. Using Program Autofinder, we got 31,000 autogroups, and only 14,000 of them had Sicilian genes. We analyzed those autogroups in various ways, such as selective pressure analysis and gene birth and death analysis. As a result, we got autogroups with positive selection, and these autogroups didn't show any geo-enrichments, but we found genes involved in organ morphogenesis and development in these autogroups, such as eye morphogenesis. The next group is autogroups with changes in gene numbers. These autogroups showed geo-enrichments, particularly autogroups with gene expansions showed enrichments in terms associated with axillary reductase activity. And final group is Sicilian-specific autogroups. These autogroups showed enrichments in terms associated with detection of chemical stimulus involved in sensory perception of smell. All these results shows adaptations to fossorial lifestyle. And another adaptation for fossorial lifestyle is limblessness, and this trait is shared with snakes. But before going to the next step of this presentation, I need to tell you about the awesome article written by Kwon and co-authors. They show that snake enhancer, ZRS, has specific deletions. They put that enhancer into a mouse genome. In one group of mice, they put just snake enhancer. In another group of mice, they put enhancer that was cured. So they put mouse sequence into that deletion. And as a result, first group lost their limbs. Second group was cured enhancer, didn't lose the limbs. So they show that ZRS is really crucial for vertebrate limb development. And we decided to study that sequence in Sicilian genomes. To do that, we extracted a region between 5th and 6th exons of a gene called LMBR1. It was shown that that gene actually has ZRS sequence in that region for other vertebrates. We used mouse sequence as a query for BLAST search, and as a result, we got no BLAST hits in Sicilian data. Probably Sicilians had the same deletions or a little bit different deletions in that region, and the lack of functional pressure and the amount of time made this region unrecognizable for BLAST search.
In conclusion, so we have produced 3 Sicilia high-level quality genomes. A large portion of these large genomes are repetitive, as we would expect. We don't see a lot of synteny in the chromosomes between the Sicilians, at least considering the Busco genes. We see that from patterns of gene gain and loss, and also from positive selection in protein coding genes, that the genes involved in all of this are involved in the observed phenotypic changes that the Sicilia present. And most importantly and interesting, limblessness in Cecilia and snakes seems to be evolved independently, but they do seem to be underpinned by the changes in the same enhancer element, which is ZRS. So we want to thank you all for listening. Obrigada. And we want to thank all of our supervisors, people involved in this, all of the institutes, Nottingham University, the Natural History Museum, the Sanger, the VGP, and the Darwin Tree of Life. And thank you for listening.
Well, welcome back to the panel here. That was a very interesting and nice set of talks. I'm really excited to see the work that is coming out of these vertebrate genomes. Really exciting work, folks. Congratulations, and I'm looking forward to more. So I will go ahead and field the questions here. Ron and I will go back and forth. This first one that's rated up is Andreas Spenning. Question to Inez. Do the implicated coat color SNPs impact the protein itself, the regulation of the gene, or some other functional element?
Yes, so we managed to pinpoint these two SNPs, which are actually consecutive mutations to the protein coding sequence of this receptor, and it is actually in a very conserved segment of the protein, so it's highly likely that it will impact the protein function. And previous studies that have shown that mutations in the same region are involved in alterations of the melanin deposition. So we are hoping to do some functional work to better access the impact of these SNPs in the protein function in these animals. Yeah.
Right. All right. Do you go by Inez or Inez?
Inez. Yeah.
Inez. Okay, I'll make sure I get that correct. All right, pick a question.
Hey. Yeah. So the next question goes to Simona from Mark Palmada Flores. Thanks for the presentation. He wanted to ask if you looked at the missing genes and evaluated their GC contents to assess whether the fact that it's missing is caused by a true gene loss or by the GA biases in PacBio HiFi reads.
Hello, thank you for the question. And this is one of the next things that we are planning to do. We didn't get the chance already to look inside this problem, let's say, but we are going to present some results from the HiFi with the HiFi data also on the next PacBio meeting. So hopefully we will have those results from that meeting.
Okay, fantastic. Next question also goes to Inez from Laura Lacolina. Which validation and quality checks did you apply to the museum sample? And did you experience any particular problems with that sample?
Hi, thank you. So for the museum samples, we did use lab protocols that are specific to these samples that allow us to take into account the cytosine deamination that is usually present in the DNA of these samples. And then during the bioinformatic pipeline, we also removed the C to T and G to A mutations that are also associated with this post-mortem DNA damage in these samples. And with these validations, we managed to get a quite high endogenous DNA proportions of about 80% to 90%. So we had no particular problems using the samples, yeah.
Okay. So there aren't, go ahead, Ron.
Yeah, while waiting for questions to come in, I had a question to Elisa, I think, about the sea turtles. So I thought maybe the variants that you found could be a difference between coming from private variants that are because you were comparing one species, one genome from one species and one genome from another species. So do you have any plans to validate the variants in more genomes would be my question.
Yeah, thank you for your question, Erin. By now we are the next steps of our research will be more focused on comparing these two genomes with other sea turtles genomes from other species, and also comparing these two genomes with other turtles from outside the sea turtle lineage. We don't have still plans to make the comparison using other individuals from the species Chelonia and Dermochelis, but it could be, of course, to comprove this variation that we are seeing that, as you said, is from one individual, right, on one genome. So it's not in our immediately next steps, but maybe in the future for sure.
Right, great. I remember those turtle genomes were some of the most complete ones that came out, so shouldn't be missing too much there. So next question from Peter Holland to Marcella, and actually Marcella, I'll follow up with the question of my own as well. So what do you think the enhancer loss why do you think the enhancer loss underpinned the limb loss? You know, what's the basis of that evidence? Perhaps an alternative view is that some other mutation caused the limb loss, and then the enhancer was a secondary change because it was no longer needed.
Yeah, that's an interesting point, Mary. I'd like to hear your thoughts because it's the same roof, let's say, for another clade, right? For another for its snakes. So they would need to have occurred twice, I would say. I mean, the loss of the enhancer function because of another mutation. But I don't know. What do you think, Mary?
Yeah, I think it's probably a lot of the evidence for this comes from the fact that this is convergent. And so we're seeing it multiple times in different places on the tree. Also from the studies in mouse, you can see that when you do your CRISPR analysis and you put the snake into the snake version into mouse, you get limb loss, but you can resurrect the limbs by putting the mouse version back in. So it seems very specific to that region, whether the mice can develop limbs or not. And of course, then as well, you've got very deep diverging species of snake like python, and they still have vestigial hind limbs that are buried in their muscle at the latter end of their bodies. And they have a small amount of this enhancer still sort of present and intact in their genome. So they're almost like a transitory phase, and I hate to use that word, but they're almost like a transition from limbs to total loss of limbs.
Interesting. And my follow-up question to that was, thinking, have you found all the interesting or evolved genes? Because it looked like the annotation found fewer genes than you were expecting. And I'm wondering, was RNA-seq used, or was this a annotation based upon homology to other species only?
I would need to confirm that. I need to confirm that.
Maybe V would know, right? But who's not here? Okay.
I mean, yeah, the annotation was done by the NCBI. So we used the annotation that was there after we submitted the genomes. For what I remember, I was looking at it, one species at least didn't have RNA-seq. So it's very possible and likely it was by homology, but don't take my word on that just now.
No problem. All right. Eran, do you have any other question you want to follow up on?
Maybe I could ask Yafei if there were any future plans for proceeding from your findings. What's gonna be your next study plan?
So this is a good question. So what I'm working on now, we extend the primate genome and to the our monkey and to the very old ancestral New World monkey and the old monkey, and we want to see how this structural variation evolve from monkey to apes, and also we are profile the single cell RNA-seq for the brain tissues, and we want to see how this structural variation variants affect the genes expression in brains. That's one part, and the other part is we cooperate with Eric and trying to build a complete T2T level app genomes for next step.
Yeah, and following up, I was wondering if the ILS is bigger in the repeat regions you were saying. Is that because you're finding ILS, incomplete lineage sorting in actual repetitive DNA? Or the genes next to the repetitive DNA?
So I think the IRS, we found more IRS on the AIR1, but we still don't know the biological meaning there. Yeah, that's what I know. Yeah.
Okay. All right. It's amazing that time. Go ahead.
But nothing. I haven't figured out. I'm still thinking about your question.
Okay. All right. Well, if you come up with a separate answer, you can email me as well and just let me know.
Sure.
But it's amazing that we're already 50 minutes into the hour, which means this starts the coffee break. This was a lot of rapid fire science. And once again, I'm really happy and I'm going to say proud of you folks, for generating such great data, from these new high quality genomes. And thank you to my co-chair, Ree. Do you have anything you want to add?
Not really. Thank you everyone again for joining us. I hope you enjoy the rest of the conference.